Hello everyone. In the previous video, we had seen how intracellular pH is regulated. So in this video, we are going to talk about how extracellular pH can be regulated by a cell, especially in the stomach, where we have very high pH in our stomach lumen, and we are going to talk about which cellular pump or channels help in that. So as you would have guessed, there is a proton pump, which is a proton potassium pump. Now, this is a P-type pump similar to our sodium potassium pump, which we had discussed in previous video. Now, when I say similar to the sodium potassium, the alpha subunit is 65% similar between the sodium potassium pump and the proton potassium pump. So now let us look at how this pH is maintained. So in the cell, we will have water in the parietal cells, and then we have the stomach lumen where proton needs to be sent. And as you see, it's already at pH 1, so there is a lot of proton there. So the water can break down to give a proton, and this proton will then be pumped out of the cell into the stomach lumen, for which we have the proton potassium pump acting. So the proton will be pushed out of the cell, and as you saw, this is an exchange of positive ion. So positive ion of potassium will be exchanged for the proton, and it will be brought into the cell to maintain the electric gradient, the charge gradient. Okay, so the proton enters the cell, and this enters the stomach lumen, and that will decrease the pH inside the stomach lumen, outside of the parietal cell. Now, along with it, to maintain the charge exchange, there is also a chloride ion which travels across its gradient and forms hydrochloric acid in the stomach lumen. And this happens through chloride channels like CIC or CFTR. You might remember CFTR from the cystic fibrosis genetic disorder. Now, as I have said, there is a decrease in the pH in the stomach lumen, which also means there is an increase in the pH in the parietal cells because it's sending proton out. And to balance that, there is carbon dioxide brought into the cell, and as water had broken to proton, the other part will become this OH minus ion, and this reacts with the carbon dioxide. Although this reaction is very slow, so we need small help from an enzyme. So Ca stands for carbonic anhydrase, and this helps in converting carbon dioxide and OH minus ion to bicarbonate ion. So it's an enzyme, so it just fastens up the process many thousand-fold. And then this bicarbonate ion can be pushed out of the cell in exchange for a chloride ion. So this is an antiporter, and this chloride ion can again be sent out of the cell to make HCl in the stomach lumen. And now we have seen that there is an exchange of proton, there is exchange of chloride, and then there is potassium ion entering the cell. And as you know, cell already has a high amount of potassium, so any excess potassium that is entering can be thrown out of the cell. There are other channels like sodium potassium pump and other channels which are also at play, but I'm not going into that detail because our key focus is just proton potassium exchange. So it happens that because of there is a lot of proton inside the cell already, a lot of potassium already in the cell, the potassium gets sent out through a potassium channel. And as you know, the proton potassium pump is a pump, so it will need energy to work. That energy comes from ATP, which is usually available in the cell, of course. So ATP breaks down to ADP, and that gives the energy to send proton to stomach lumen, which has lower pH, which means more proton. So from lower concentration gradient, we are sending it to a higher concentration. So we are sending it against the concentration gradient, and ATP is used, while the chemical gradient of this potassium is maintained through the channel, and then there is also sodium potassium pump to play at different parts, which we'll not talk about in this video. Now there is a lot of all these channels and pumps. Their activity is highly regulated by chemical factors like hormones coming from other cells, and we are not going to talk about that a lot, but we'll gently touch on that topic, the chemical regulation, how it happens, or at least how chemical cues or signals regulate through proteins called GPCRs, G coupled receptors, which when the chemical signal comes, it can induce the acidification of stomach. Although there is a basal level of acidification always on in the stomach, usually only when a signal comes, we have more acid secretion. And we are going to talk about the structure and how the structural changes in GPCR affect this function. And just to let you know, a lot of disorders which happens, gastrointestinal disorders that dysregulates acid secretion, happens at the chemical cues level, so they affect the chemical signaling pathway. But we'll talk about those. We'll not talk about the disorders, but we'll talk about GPCRs in later video. Here I just, in this short video, oops, let me make it smaller. In this short video, I just wanted to let you know how these pumps and channels and antiporters exchangers, they come together to control extracellular pH through HCl secretion.